Okay, welcome back again. So, now we finished our origin of angiosperms, origin of flower. Okay, now we are moving on to part by one. First one is origin of stamen or the evolution of stamen. So, how stamens, they are the male reproductive part of a flower. How the stamen evolved. So, we can look upon to different concepts regarding the evolution of the stamens. Okay, so before going to that, a brief introduction regarding the stamens. So, stamens are usually a slender organ, and basically the rank of the stamen is leaf. You already know flower is a modified shoot. Okay, and they are closely parallel to the organs in the morphological and anatomical structure relation to the stems. Its origin, while you look upon ontogeny, they are all in relation to the stem. That is a floral receptacle, thalamus, So that is why it has close affinity to the stem or leaf. In most of the families, stamen is highly specialized and great variety of different forms you can see, and these modification of stamen are basically related to the type of pollination. Clear? And a typical stamen having of two parts. What are they? Filaments and anthers. So filaments are the basal sterile part, and anther is the apical fertile part. And anther carries the microsporangia or maybe sometimes microsporangia or pollen grains, and as together with some sterile tissues also. You studied in embryology the detailed structure of the anther. Okay, so clear, here you can see the picture of the anther. That is a filament, this is the anther, and these are the four chambers or microsporangia, okay, that carry pollen grains. Okay. Then one more important part of the stamen is the connective. Connective are the small strip of tissue lie between pairs of sporangia. This, see here, this tissue is the connective. Okay, and not separable histologically or morphologically from tissue of anther stalk wall. Okay, for both are same. And it may sometimes extend beyond the sporangia. Some cases that connective can be appear up to this. Studied in the degree classes and non-ACA members, it appear like a hood. Okay, and filaments and connective are and appendages of connective constitute part of the microsporophyll. They are not morphologically distinct. They are the part of microsporophyll. So that's some modifications in the connective part of the stamen. So our question is, what is the structure of a primitive stamen? Clear? So now up to this we discussed what is the stamen present in nowadays in our flowers, that you are familiar. But our main part is what is the nature of the structure of the primitive stamens. So look in that picture. This is the primitive stamen that appear like a leaf. Clear? Leaf. So you know that fertile part or spore bearing leaf is known as what? Sporophyll. Clear? So this is actually the spore, sporangia or pollen grain. That bearing leaf is actually what? Sporophyll. So you can say this is a sporophyll. Clear? So this is a leaf bearing sporangia. But what happens later? You see the sterile part. So here huge area is sterile. So only this two part is fertile. Clear? So sterile area is greatly reduced, condensed, and finally that became the anther like this. Clear? So that is the most, and one example is the most primitive stamens you can see in the ranales, and they are the broad leaf like organs. Clear? Okay. So in the primitive stamens there is a clear cut distinction between the fertile and sterile portion. Understood? And that is appear like a leaf that condensed and reduced and finally became only the fertile part, that is the present stamens. Clear? Okay. So sporangia born usually at the center of the sporophyll. See here on the abaxial side in many of the family like Degeneraceae, Annonaceae, etc. So in Degeneraceae and Annonaceae, so these are the very some primitive families, Degeneraceae and Annonaceae. You can see this is stamens usually occurring on the abaxial surface. This is a leaf, it's abaxial surface, and Some cases in Ostrobalia and Magnolia, it occur on the adaxial side. So, two surfaces are there, one is abaxial and one is the adaxial surfaces. Okay. So, anyway, this is the condition here. So, when you cut it, cross section, so cross section like this, cross section you can observe like this, arranged. So, when you just go back to your pteridophytes class, fern classes, see, okay. And this is the members of Anonaceae. See what is the structure, sterile condition. When you look, compare this with this, the nature of the stamen of Anonaceae. This up to apical portion, sterile portion, middle portion only fertile. What is seen here also? Same. That is reduced size reduced in Anonaceae. So, okay. So, that is the nature of evolution. Clear? See, this is another very important one. Stamens with semi-staminal form. See, that modified petals, modified and finally become the stamens. So, these type of conditions occurring in the Ceratophyllaceae, Nymphaceae, Eupomasiaceae and Leduridaceae, etc. And Lacibalaceae. These are all some of the common modified semi-staminal forms. So, sterile portions. In the last slide we studied this fertile part here, okay. So, what happens here, on the lower portion complete it becomes sterile and this is concentrated on the apical part and gradually that sterile portion that become the filament and finally into the stamens, okay. So, specialization means what? That is advancement means what? Reduction of the sterile tissue, clear, okay. And reduction of the marginal area. So, this is the marginal area, clear. These two sides are the marginal area. So, that is going on reduced, okay, and finally become the fertile stamen. So, lamina is progressively narrowed, you can look upon. So, this is the broader region. What about this? This is a narrowed one. So, lamina is narrowed, okay, and proximal part become filament. What is the proximal part? This is the proximal part. What is this? It become the filament. Okay. So, lamina is narrowed and proximal part become the filament and apical part become the fertile. What about these sterile portions? They are greatly reduced and lost. Clear? So, the median region, this is a middle region. What is the appearance of this middle region? That becomes the connective. Understood? Okay. So, this is the way how they are sterilization taking place and how primitive to advanced. Okay. And some cases you can see this is an sterile region. So, again the sterile region persists here. What is the fate of the sterile region? So, that become a hood like structure. Okay. See here. See in Anonaceae, sterile region become hood. Okay, so these are all stages you observed in some of the families and members like Ostrobalia, Magnolia, etc. and Nymphaceae and so on. Clear? So, this is another one more picture to showing the trend of evolution of stamens from primitive broad laminar type, Thaktajan, according to the view of Thaktajan. Okay, you have to draw all this diagram while discussing the evolution of stamens, and these diagrams and photographs also may ask for your practical examination in the morphology part. Understood? Now, about the anther structure, see different types of anthers are arranged. Great variety of forms are there. They are usually based on the attachment of the filament, basifixed, alternate, dorsifixed, then versatile, like different forms are there. So, lateral arrangement of connective makes the lobes apart from each other, two anther sacs. See, form of connective determines the form of anther. If were connective, basifixed, connective at basal part, alternate throughout connective, dorsifixed backside, versatile just middle side. So, how this connective touches or fixes, based on that you can have different types. So, primitive family constitute a major part of anther. So, in primitive family, sporangia are minor structures. Okay. Advanced families, what is the condition? Connective is very slender median axis. Very example is Graminae, Poaceae. So, connective is very slender, attached just as a thread, point of attachment to the anther lobes. Clear? And a series of reduction occurring in from massive connective in Magnoliaceae, then Trocodendron, Tetracendron to a mere fragment in Graminaceae. See, that is the difference here. So, while you take an example of Annonaceae itself, you can see connective is very thick and protrude out like an appendage hood. Clear? From that type of anther, connective, it is gradually reduced and become very narrow, very thin, very thread-like structure, just an attachment to the middle part of the anther that is occurring in the graminae. So, see the evolutionary trend in the connective. Clear? So, in sometimes you can say this connective may be absent in the graminae. You cannot differentiate connective here. You can see only the filament here. Okay. Then proximal region, proximal part of the connective is merges with the anther. Okay. Distal appendage of connective are characteristic to many families. Many of them are having distal appendages. See, this is a distal appendages, but proximal appendages are very rare. So, reduction in the distal appendages reduced and that is observed only in primitive. This is the distal appendage observed in the primitive family, that is gradually reduced in advanced family. You cannot see such appendages. See here, you can't see the appendage. Clear? Now, filaments. So, filaments case, in the case of filaments, you can see primitive filament means flat and expanded. But already told, you cannot differentiate here a filament and anther. So, this is a flat structure, expanded. But when you look upon to the advanced filament is very thin. Looking into the filament of Graminae or Poaceae, very narrow, thin structure attached at the middle portion of the anther. So, that is slender and stalk-like. Okay. Now, so relationship, this already we discussed. Now, evolutionary trend. Androecium is segregation of stamen of a flower. Okay. They are arranged spirally, stamens in spiral. These arrangements you can see. Spiral arrangement is a primitive type and whorled arrangement originated from spiral arrangement. So, spiral is the primitive type. Where you can see this in the primitive family like Annonaceae, Ranungulaceae, Magnoliaceae, etc. The spiral arrangement of stamens are observed. And most primitive type is observed in the Degeneria, Austrabalia, Himatandra and Magnolia. I already told you. The laminar three-veined leaf-like structure without a fertile and a sterile part. Sporangia on the near the center on the abaxial side or adaxial side. That is actually repetition. That is I concluding that part. Now, semi-laminar stamens also observed in Upamaceae, Ceratophyllaceae and Nymphaceae. You have to remember and draw diagram also. Then further specialization, what happens? Reduction, what is a broad laminar reduced to narrow. Apex may be sometimes modified appendages. Then center part become the connective and basal part become narrow filament. Okay. Midvein region, connective and distal part appendage. Clear? Now, primitive family, connective forms major part of the anther. So, this is the primitive family, connective is the major part. But if you look at the advanced family, you can see the highly reduced connective or connective may be absent. In Acanthaceae usually and Plantaginaceae, highly reduced connective. Okay, highly reduced or connective observed in certain families or may be absent. Betulaceae, connective as well as upper part of filament divide into anther lobes. This is the connective and upper part divided. Okay, most primitive family connective reduced into an appendage. Disappears when it become more advanced families. Now, broad laminar filament merging with the rest of stamen. See, microsporangium into stamen, narrower and it finally become advanced family. So, this is a diagram and drawn diagrams. You can look into and different modes of sterile appendage left. Okay. Stamen with a narrow filament, basal dorsal versatile attachment, basic fixed condition and versatile advanced, basic fixed primitive. See, how to look it, basic fixed is what? Primitive and think about versatile in Graminae is considered as advanced. Clear? These are the four types. Now, so most primitive stamen, one concluding most primitive stamen is broad, laminar, differentiated into the vein region, not into filament and anther and connective. Clear? Okay. A continuation of connective beyond anther like a sterile tip, primitive feature. Stamen become more slender and laminar, disappearing, and sporangia occupying marginal position. See some different evolution stamen. Primitive laminar stamens without a clear distinction into anther and filament. This is the one. But second one, Ostrobalia, adaxial pollen sacs in Ostrobalia. See leaf-like structure, adaxial pollen sacs. Okay. Hematandra, abaxial pollen sacs. Abaxial pollen sac. This is adaxial pollen sac. Only the part of the leaf is vary. One upper side, one lower side. Okay. Then Degeneria with abaxial pollen sacs. Okay. Magnolia with adaxial pollen sacs. But see in the magnolia it is reduced from this into this. It is sterile portion greatly reduced. Clear? And that sterile portion and fertile portion move on to the margins. Then laminar stamen of magnolia in this condition. This is a laminar stamen of magnolia. Then semi-laminar stamen of Michelia. This is semi-laminar stamen. See, almost all sterile part gone and origin of the filament started. This is the magnolia, laminar stamen, semi-laminar stamen of Michelia champaca in Magnoliaceae. Clear? And outer semi-laminar stamen of Nymphea. What is that? This is one. Outer semi-laminar stamen of Nymphea. And inner stamen with a narrow filament. This is a filament of Elysium, stamen of Opentia. Clear? And this is stamen of Poa. That is advanced one. And L, this is stamen of that is penstemon with a well-defined filament and large anthers, distinct anthers, filaments. So, see the evolutionary, our steps of evolution. This is the first one. What is that condition? Broad, with fertile portion only, small portions. Okay, so upper and what about this? Some another stage. See, somewhat in the center, this become narrowed and this part will start to become filament. This part may be degenerate or become hood. What about another peculiar type? Another type of somewhat similar to this. This is also somewhat similar to this. This is another very important. You can mark it as third, magnolia. This is also, you can mark it as third stage, that is Michelia. See, sterile portion move at the center only, fertile in the both sides and base also become filament. This is another important part, and this is a semi-laminar, another step, semi-laminar, and this is the somewhat most of the, you can mark it as 4, become the fertile portion only on the upper side, and this is the perfect stamen, that 5. Okay, so these are the somewhat evolutionary stages going on in the evolution of stamens. Okay, so very important, laminar, leaf-like to how that present condition of stamen occurred. So very important to study. So once again, so whorled arrangement is a modification. I already told spiral is the first one. When stamen in one whorl, that is haplostemonous. When stamen is in two whorl, that is diplostemonous. Outer whorl alternating with the petal and inner opposite to petal. If it is in reverse condition, outer opposite to petal and inner alternate, that is obdiplostemony. So haplostemony, diplostemony, obdiplostemony. More than two whorl, polystemony, that is very rare case. Then diplostemony is the primitive type. Others evolved from diplostemony. Obdiplo is the very less common, occurring in Rutaceae. Obdiplostemony is, you can see, outer wall opposite inner wall, and also Rutaceae I studied in UG class, and now you can have Caryophyllaceae and Oxalidaceae. So Oxalidaceae and Caryophyllaceae shows obdiplostemonous condition. Okay. Now, haplostemonous is typical in Asteraceae and Lamiaceae. The cyclic antration contains one whorl of stamens only. Ophaplostemonous, another one, stamen opposite petals. Only stamens opposite petals. That is Balanophoracea, Whiteaceae, Sandalaceae, nothing in your syllabus. And both originated from diplostemonous condition. So diplostemony is the, see, obdiplostemony is very less common. Diplostemony is the primitive. Clear? Then all others derived from diplostemony. The reduction in androecium, individual organ or wall may be lost. That is also very important. Obdiplostemony is an union of two stamen walls, shortening of receptacle. That is a reduction. that is two whorls occurring in obdiplostomy, that may greatly reduced by fusing the two stamen, one alternate to petal, one opposite to petal, that become fused. So likewise it will get reduced. So reduction is an advanced feature. See, alternate opposite to petal, alternate to petal, opposite to alternate. So maybe greater reduction taking place. That's an advancement feature. One more reduction is stamenodia formation. So specialization into fertile stamen into sterile stamenodia. This is also very how sterilization take place. Fertile stamens modified into sterile staminodias. Okay. Usually occur in inner or outer member. Outer wall becomes staminodes, inner wall become fertile. So you can see in most of the monocots cases you studied in Zingiberaceae, Orchidaceae, and all staminodes are present. Okay. Transformation of inner stamen is more primitive. Inner stamen seems to be more primitive condition. In some member Degeneraceae, Nymphaceae, etc., staminodia present both above and below stamens. That is a starting of evolution. Okay. So, two conditions are there. One is the in the obdiplostimony fusion of whorls. So, one whorl become fused and reduced, or some cases what happen, fertile stamens become sterile stamenodia. So, these are the reduction in the during the evolution of stamens. So, reduction in the androecium where the spiral whorled is taking place into stamenodia. So, you can see these are all staminodes. See, these are all staminodes and these are the fertile stamens only. Clear? So, staminodes present in staminodes here absent. This is the first condition, this is a reduced condition. So, that is occurring in it's a very common feature. Eupomacia is an example where petaloid staminodia from prominent corolla part. So, these are the actually petaloid staminodes that form prominent corolla above and inside the androecium. Then Degeneria, Cyclanthus, Nymphaceae, etc. Staminodia present both above and below stamens. You studied that. Above and below stamens, staminodia present. Then monothecous anther result from splitting of stamen, separating two anther lobes. Splitting of stamen, separating two anther lobes. Then conation of two stamen result in dichotomy in salix. These are all how fusion take place. That's an example here. Monothecous anther forming by splitting of stamens resulting two anther lobes. So, I hope so we gone through the different steps. So, while discussing the evolution of stamen, you have to first explain what is a primitive stamen, what is the structure of a primitive stamen. So, by taking different examples, you have to draw the primitive stamen, its nature. Then, what is the process of going on? What are the changes occurring in the anther part, in the connective part, in the filament part? Then different types, semi-laminar stamens, then appendaged one, then reduction, spiral into a whorled condition, diplostimony into obdiplostimony, that likewise, and also you have to note down the staminodes, fusion of the fertile stamens, and also reduction of the or sterilization of fertile stamen into staminodes. So, anyway, a number should be reduced. Okay, that is the advanced character. Okay, sterile portions greatly reduced to fertile portions. Clear? Broad sterile portion reduced to fertile portion. In the fertile portion, the connective like structures are later become very much reduced. See, so likewise we reach the advanced stamens of the like members of graminae. So, clear? You have to draw all the diagrams, you have to quote examples and explain how evolution takes place in stamens. So, I hope you understood well, and also very careful, these diagrams may also appear for your practical examinations. Okay? So, have a nice day. Thank you.